context. So in the first talk of the workshop, Keith Wiley was explaining to us why our memories are the essential thing that makes us who we are, or at least the thing that makes us, individuals as opposed to just standard generic human. He explained that the so-called connectome and some additional information contained in the biological machinery of our brains contains the traces of all of those memories. And he explained that this is why efforts at mind uploading focus on using that information to create an emulation in an artificial brain that constitutes the uploaded person. In our second talk, Ken Hayworth explained that in more detail, and also the Brain Preservation Prize, motivating the development of brain preservation and preservation procedures that maximize the likelihood that all essential memory traces are preserved. And we tried to talk about some of the critiques that might come up. Then before our short lunch break, Alicia Smallwood went through some clips demonstrating how Ken Sharif and others go about the process of reverse engineering and integrated circuits, which is a domain that is a bit more mature than brain reverse engineering and has some similarities with it. Although Lou Sheffer, who's here, pointed out that one of the big things that they take advantage of is that, of course, these are design systems. And so it's a lot easier for us to imagine what's really going on inside of each one. We can use that to improve our odds of being able to reverse engineer what's there. And that's somewhat different than a biological system. Then Jonathan Gornet presented his published work on reverse engineering the workings of a small part of Drosophila brain based on knowledge of the connectome and prior information about the expected neural responses. His work is probably some of the closest that anyone's gotten to restoring brain function from a preserved brain. There are a few other examples out there of attempts to go do that in a few other animals, but this is very much to the point what Jonathan was presenting. And the challenges where extra effort has to be made to produce a usable model were very instructive for the challenges that we should expect every time we attempt to recreate the mental functions of a specific brain. Now in this talk, I'd like to focus a little bit more on that aspect. So let's see if I can get the slide to go. There we go. So we're back to this panel here, and I've highlighted the pieces that I'm going to be talking about right now. I'd like us to take seriously what information and what understanding are needed to make a brain model work, and to make sure that the brain model works as it should when the goal is to, say, for example, restore a person. I'd like us to think carefully about where that information and that knowledge can come from. Which information can be plucked out of the preserved brain itself, which knowledge we can provide by having a general understanding of human brains, and which information unique to the individual has to be supplied in addition to the preserved brain, which is something I think maybe Ken is going to give me some pushback on. But anyway, stick with me. So the title of my talk is Model Validation and System Decomposition at Multiple Scales, two aspects of the process that I want to emphasize, because they both have direct impact on the information that needs to be stored so that a person can actually be brought back. At least that's the claim I'm making here, or the detail I want to point out. Now, I'm going to take another look at that IC reverse engineering here because I think there's something of relevance in there. In 2009, Randy Torrance and Dick James wrote a comprehensive and very readable paper called The State of the Art in IC and Reverse Engineering. And in it, they make this important observation. They say, the analysis phase can be very iterative and use many sources of information. Often public information is available for devices. This can take the form of marketing information, data sheets, technical papers, or patents. These can often help with the schematic organization, for instance, if block diagrams are available. They can also help in the understanding of architectures and sometimes circuit designs. And functional and timing analysis can be further validated using simulation. Multiple methods are usually used for verification. Note that word verification there. We can compare the process of building a brain emulation for Jane Doe from the preserved brain of Jane Doe with the reverse engineering of a specific brand and version of a microprocessor. Take, for example, and this is one that Ken Sharif also already mentioned, the 8-bit Zilog Z80, so very famous, shown here in images from the Wikipedia Commons. The version above is from 1976 and the one below from 1990. All programs written for the Z80 will have to work on an emulator that's built for that. Of course, it's also useful to know a lot about CPUs in general from the 1970s and 1980s, to know the typical circuit logic that one tended to implement on those chips, to know how that was accomplished with numerous built-in tricks for efficiency. Understanding the MOS 6502, for example, also from 1976, can be very helpful at the system level. Understanding one of the successors of this Z80, like the 32-bit Z380 from 1994, likewise. Still though, even then you still need to validate the emulated Z80, our Jane Doe, who we're trying to reconstruct and revive. On the emulator, we want software to behave as if it were running on the Z80, just like we want the characteristic behavior of Jane Doe. By the way, and just to take this analogy to its extreme, as it happens, the Z80 was a software-compatible extension and enhancement for the Intel 8080, another famous processor. In this sense, the Z80 itself has to be a reliable emulation platform for the Intel 8080 software, which is an apt metaphor for the desire to be able to move mental functions to a brain that can be enhanced and extended. Now, keep in mind that methods for estimating sets of parameters in many cases cannot lead to a single optimal solution. Instead, they explore a solution landscape, which I'm trying to sort of depict here on the left-hand side, which has many local optima. Which combination of the possible valid solutions for each of the estimated parameters produces Jane Doe, at least to the degree of the success criteria you set forth? How can we determine the degree to which we're restoring her? Could we develop a dynamic fingerprint for Jane Doe with which to compare restoration candidate Jane Doe number N, for example? Oh, that didn't work. Let's see, sometimes I'm clicking in the wrong place. Now, a practical and hopefully successful restoration process will almost certainly involve an iteration of model selection, parameter search, model validation, and corrective modifications, then going round and round for a while. This is something I just really want to emphasize, that this is a process. It's not just a single, you just don't go and build a model and then it's done. And this is very important because we need to make a distinction between theoretical, what we can theoretically hope to get from a data set, and what we can practically do. To do better than a brute force search of the search space, we'll want to learn in each of these iterations. And on average, we want the match to improve. Given the high dimensionality of the search space, there's a combinatorial explosion of possibilities. Imagine if the estimation of each parameter merely involved solving a simple quadratic equation with two possible solutions. Wherever these choices may be treated independently, you'd find an explosively growing binary tree like the one that's shown on the left here, which only has 10 steps, actually. If you had 1,000 independent choices, that tree would be something like 10 to the power 301. And just to give some idea of how big that number is, there are only about 10 to 78 or 10 to the 82 atoms in the universe. So this just shows that, the problem of the search space is a real one. The explosion of having to carry out a difficult search when you're estimating or optimizing is often the death knell for any modeling of a modeling process that you have. You want to avoid that at all cost. So we probably need our validation results to tell us more than just how much the dynamic activity matches the Jane Doe fingerprint. We'll want to have cues that correct and intelligently guide our search algorithms. So what are these cues? Where do we find them? Do we find them in the preserved brain, or in the dynamic activity generic to human brains? Or do we find them in the dynamic activity of Jane Doe's brain? Or perhaps they're distributed throughout those different sources. I think that's very likely, actually. So some, you can pick up traces or evidence of them in different origins or sources of information. Some are here, some are there, and some are, they exist at different scales and resolution in there. And which one is the best one to pick may depend a bit on the tools that we have available. Cases where the necessary knowledge to guide some aspect of the search could be deduced or inferred from more than one possible source of information are shown here in the overlap of these sets. And in those cases, you'll want to know which of these sources of data is most easily obtained. Is this a case where it would be easiest to just record something from that individual brain before storage? Or would it be easier to try to infer from the preserved brain? Or would it be easier to take something that's in a generic data set, generic activity, generic knowledge of how a circuit works, and use that to constrain? Or are we then losing some detail that we actually needed to pay attention to? So in any case, we were talking about this problem of having this gigantic system where we're trying to carry out a whole bunch of parameter estimation all at once, and model selection, not to forget that. And of course, what you do is you go and decompose the system into smaller bits. The sensible decomposition of a large unknown system is something that, of course, IC reverse engineering also does. In fact, if you paid attention to what Alicia was showing us, one of the things that Ken Sharif does is, he'll go in and he'll try to identify different parts of the layout that appear to represent functionally distinct pieces before he tries to carry out any sort of reverse engineering. Then he attempts to reverse engineer each one of those, and having done that, he composes the full reverse engineered system from all of the separate parts. Now, an advantage that he has is that we know that the entire system has agreed to speak to itself in a specific way. It uses the ones and zeros, the high voltage, the low voltage in a certain way. It will have a clock timing activity. So there are ways to make sure that the whole thing doesn't break down when you integrate all these recomposed small pieces that you've reverse engineered. That's knowledge, intrinsic knowledge about these systems that you're putting in there. It's like having generic knowledge about how human brains work and how the different regions talk to each other, and trying to use that to constrain what's going on, but not just entire brain regions, even lower than that, smaller than that, tiny neural circuits. How do they manage to work together, and how does it not break down if we're reverse engineering them separately? So we're gaining some things and we're losing some things. After decomposition, it may be easier to produce plausibly human results because we can constrain each subsystem at every scale in accordance with our understanding of generic human brains. But then the composition means that you begin with lots of subsystems, each obtained from a set of possible solutions, all working in conjunction, so they need to do that correctly. And then just to remind ourselves of what I was already talking about earlier today in the introductory talk, there's this issue of compounding errors. Without validation at granular levels, if you don't have validation data at that level and also at successive composite levels, then you're likely to end up with problems like this. You might end up in cases of intractable compounding errors where it's hard to find out exactly where things went wrong, because things could be going wrong in one spot in that chain or in multiple spots at the same time. So the debugging becomes really hard. Okay, a little bit more about small subsystems. This is, of course, obvious to most of us. When we take the same toy example that we had just before, and we say, okay, we had these 1,000 binary choices. Now, if it's possible to cut this system up naively into 250 subsystems, each of which you only have to make 4 choices for your estimation among 4 different possibilities in each case, then you end up with something like 4,000 different possibilities, much smaller search space than what we had before, which was basically infinite. In fact, when I typed in that number, I calculated R there on Google, the calculator there told me it was infinite, which I disagreed with, but I guess it's close enough. So this is a lot less than the previous number. That's better. Now, so it seems like we certainly want to decompose the unknown system into many sensible smaller subsystems. And we then apply our reverse engineering methods to each of those subsystems. Perhaps we build a cable model, a compartmental cable model of the neuron that we found in each, or neurons that we find in each. We populate those dynamic variables with our best estimates based on geometric measures and counting of components within a 3D image produced from the preserved brain. So those components that we can actually categorize, if you can categorize and identify, then you can do things like measure and count, and you can make sense of them and put them into functional models. You can do this categorization in a probabilistic way. You can attach confidence labels, and perhaps we can raise our confidence for both categories and parameter estimates by reliably identifying which typical human brain regions and neural circuits this particular subsystem is a part of, and therefore what it should be like. Now we've got very many small reverse engineered subsystems as a result. The bit about that small subsystem from the previous slide, of course, was a toy example. More realistically, you could imagine something like, imagine we're building a cable neuron model from these images of dendrites, and each measurement of a compartment comes with some uncertainty, some error in the sample. Together, that makes for a lot of choices. Are we going to err on this side? Are we going to err on that side? And do we keep on erring in the same direction every time we make a measurement? What happens when we put all of these different estimates together? This is why actually Jonathan's point about how much these kinds of details mattered or didn't matter is very important. That's something we really need to know. Anyway, so the more we know about generic brains and the better we're able to do our scans and measurements, the less we're in need of specimen-specific validation data. But at the same time, we're also going to be improving our ability to record activity. So if we're looking into the future, who knows where the sweet spot's going to be in terms of information sources for a model build and for its validation. Like every model, even at this smallest scale, or granular scale, of our reverse engineering enterprise, each model system needs to proceed iteratively through those model selection, parameter estimation, model validation, and correction stages. Now, do we have the data that we need for model validation and correction at that level? Which of our possible information sources does the data come from? If we need dynamic activity data, then behavioral data at the whole system level, or macroscopic fMRI or something like that, what you would apply to a living human, probably won't do. Dynamic activity data generic to all human brains may be useful, but will that be enough to guide validation and correction toward the particulars of how some granular subsystem was tuned in Jane Doe's brain? We have to be careful because there's a risk that comparing only with generic human data sets will tend to produce the strongest validation performance outcomes whenever a subsystem, as you're modeling it, is pulled towards the generic mean. So it's like in these Drosophila systems where basically they're all the same, it's kind of hardwired. That's the generic mean. This is what you're going towards. It's like, this is the bit we know how it works in the human brain. You're going to make it just like that. But as you're going in that direction, we want to use this generic knowledge to constrain the subsystem reverse engineering so that the subsequent integration will tend to produce healthy working human brain emulations. But in that process, we don't mean to obliterate the deviations that make Jane Doe uniquely Jane Doe. Now, so far, we've been talking about reverse engineering a system that produces a desired characteristic activity or response, the kind of response that is Jane Doe. But we also have to care about reverse engineering the first and perhaps even the second derivative of the system as well. As time goes on, we would want to verify that the emulated brain continues to respond in a satisfactory manner, while new experiences, of course, necessarily introduce changes. Those responses have to evolve. As biological brains are noisy and chaotic systems, our predictions and criteria for what constitutes plausible evolution for Jane Doe is a range of chaotic paths. It's not just a single path. The emulated brain does not need to evolve its response along one particular path that the original brain would have taken, but should do so within a range of paths if we are to deem that emulated brain an emulation of Jane Doe's brain as opposed to, say, someone else's or some brain that doesn't even exist. Here's the depiction of an example of that with fantastically reduced dimensionality and over a very short trajectory. It's like just a very short evolution of the system from one state where this white dot there, that's the snapshot where we have our preserved brain. We've built an emulation, you let it run. Those green paths are within the acceptable range, whereas, say, the red ones would not be. And how did we determine which range was acceptable? What paths are okay? I don't know. That's something we still have to figure out. But yeah, without the validation data for the first and possibly the second derivative of the system, the problem's not just one of minor deviations or some acceptable blurring of the characteristic activity. Without that, we can't even determine which chaotic systems have a characteristic evolution that can be considered a restoration of Jane Doe. How do we know it's the green ones and not the red ones? Without considering validation, we wouldn't even have specific criteria for the width of that acceptable range. We wouldn't have any specific measurement for what is success in whole brain emulation. So it would be comforting to me if I could believe that we need no recorded activity data to successfully reconstruct the characteristic activity behavior of a system. Unfortunately, my modeling experience tells me that that's very unlikely. I have yet to see anyone model a large and complex system, a biological system, where the solution landscape for the parameterized system is so straightforward that there's only one possible solution, and where that one solution is correctly discovered without validation and correction. And let's be honest, even fairly simple and small systems don't tend to work out that way. These two problems, not knowing which solution in the landscape of possible solutions is the right one, and the improbability that you'll find yourself there on the first step, those probably exist even with a total understanding of how brains work. If you store no validation data with a brain, you are probably reducing the chance of revival to a tiny fraction of its likelihood in the best of circumstances. Okay, so then, this here is important because, of course, Ken was already alluding to future science, right? And I just mentioned something about what happens if you understand the brain completely. When we have a full understanding of the neuroscience and the neural engineering of human brains, perhaps when we build whole brain models, our initial constructors and our constraints will be so sophisticated that each one of the models we build is immediately a working human brain. We may then be able to eliminate all of the terrible failure models that would otherwise have resulted. That's already a great improvement. But even then, you still, I'm just racing ahead here. Didn't mean to do that. But even then, you still need to question if a resulting whole brain emulation actually satisfies the criteria of having restored Jane Doe. Without validation data, there's no way to claim such an accomplishment. Knowledge about brain functions can teach us about dependencies and homeostatic processes. Understanding those removes many of the branches in the full search tree of possible combination of solutions. But what does it leave us with? I mean, we have to wonder about the rest of the landscape, of that solution landscape, for this set of jointly approximated equations in a realistic system. So there's no doubt in my mind that we do need to care about validation data. The question that remains is, of course, what that data needs to be. At what temporal and spatial scale, and at what temporal and spatial resolution do we need to obtain activity data? At one extreme, are we talking about 5 minutes of data from an fMRI with 1 cubic millimeter resolution? At the other, are we talking about months of neurophysiology recordings, sampling at 30 kilohertz, and listening to 50% of all the neurons? We certainly shouldn't be hasty or flippant in trying to answer that question. This is not a matter of guesswork. What it really needs is further study of the methods applicable to reverse engineering and brain model building, and a study of the strategies that can be used to guide those through decomposition, model selection, parameter estimation, model validation, correction, and composition, with iterations at multiple system levels. Fortunately, this is already underway. Here and today, we clearly and concretely identify these questions, and those researchers who actively work on reconstruction based on connectome data and who actively work on neural prosthesis are beginning to discover methods and strategies. So, if you want to assess which information you need to store to be able to successfully reverse engineer a complex system, specifically the brain, and if the success criteria are not merely to find some plausible solution, but one solution or a very small subset of acceptable solutions, then you have to care about the information that will be used throughout the reverse engineering process. You need to care about the data that support a strategy for cues to direct a search through the solution landscape. My cautionary message is that it's not a matter of better understanding the brain, nor is it just a matter of building more powerful computers. The challenge is based on fundamental and inescapable truths of mathematics and nonlinear dynamical systems. Think about it. Even if you had a magical computer made of infinite computronium and infinite speed or infinite time, so that you could generate all possible solutions, whether there are just 12 or some stupendous number, you would still not know which one is the emulation of Jane Doe. So if you want to understand Jane Doe's odds and what it would take to revive Jane via whole brain emulation, then you have to care about the process involved. That goes for both of the approaches that are receiving serious attention: whole brain emulation via neuroprosthetic replacement of all parts, or whole brain emulation via reconstruction from preserved brain. Any form of whole brain emulation depends on building a working model of Jane's characteristic mental functions. Whole brain emulation is therefore subject to all the same fundamental difficulties any computational modeling project encounters. The process involves at least these steps: something like decomposition of the whole into manageable parts for each. For each part, then, you have to do model selection for all of the dynamic components. And for each of those, you have to have kernels. Parameter estimation or optimization. I wonder if I could actually here we go. Oh, no, that just moved me forward. So I can't just, here's a pointer, yeah. So parameter estimation, then model validation, model correction. And then, of course, you can iterate back through these until you've satisfied a specific success criterion or you can't improve further. Then composition of the separately reconstructed parts. Then you validate the composed model again with validation data. Maybe that's different validation data. And again, you might have to go back, iterate back through steps all the way from the beginning. And then if you have the whole put back together again, you want to validate that whole again in some sense. Perhaps this is where you can use, very generic, large things like an assessment quiz or something, I don't know. And it's possible that you have to carry out this whole thing a number of times with different choices. So generic knowledge about human brains, better than the generic knowledge we have now, will of course help with each of these steps. And it's likely that there will at some point be knowledge good enough that every time we build a model, yes, it's a plausible working human brain that you end up with and human brain parts. Specific data, such as collected from a preserved brain or from activity recordings, is the data that helps drive you towards building Jane Doe's specific working human brain. But beware, you need one set of data to carry out steps 2 and 3 here. The model validate, I think it's actually different numbers, but doesn't matter. To carry out the model validation, the model correction, you need another set of data, a fingerprint of your mind for the comparison over here. Everywhere where we're doing this validation, we may need a different bit of data, and we may need data at different scales. So, yeah, a major issue here is that reverse engineering a complex dynamical system is much like trying to solve sets of polynomial equations. They're not going to have just one mathematically correct answer. Each one will have several or maybe many. And the composition of many equations means you have a deep tree of many possible solutions. And there will only be a small subset of those solutions that satisfy your actual criteria for having brought back Jane Doe in the way that represents how Jane's mental processes could evolve. So don't underestimate the importance of model validation and correction. The problem is not just one of gaining understanding of human brains or of having faster computers and smarter machine learning. Right to the granular level, we have an unavoidable problem of exploding choices. I mean, it might be that we can constrain it a lot, but there still are some choices. It's unavoidable. Now, we can deal with that if we know enough about the reverse engineering procedure and the data that's needed to guide it. So yes, do preserve and store the brain, but take care to store the brain's validation data at the necessary resolutions as well. In closing, let me stress that I'm not coming at this as a major skeptic of the whole enterprise of whole brain emulation. Far from it. I'm probably one of the most public advocates and promoters for this as a science. What I'm trying to achieve with this talk today is just to bring the discussion back down to earth, to ground it in the actual practical problems of reverse engineering. Sure, of course we want this to succeed, and of course preserving a very clean snapshot of your memories in the form of top-notch brain preservation is a great step in the right direction. But while we acknowledge the importance of that accomplishment, let's not be distracted too long by the beauty of that snapshot. Let's remember to keep our eyes on the actual requirements for a whole brain procedure to succeed. And that is my last slide. And I think maybe I'll switch to the camera, if I can stop sharing. Oh, here we go. So since I'm moderating myself, I'm assuming that Ken has some questions or things to say, indeed. But so do people here in the room. Let me see if we can get Ken the first word, just because I think he deserves that right now.
Do we want to also switch to the room camera?
Yeah, that's a good idea. Let me just switch that. Okay, so just 1 second. I'm still doing that switch so we can see the other people in here, and then we'll get into the questions. That's the OWL camera. Microphone as well. Oh, the microphone has to switch to meeting OWL. Yeah. Is that it? All right. Yeah. So Ken, I'm assuming that you have something on your mind.
Yeah. So
Wait, you can't hear me because I
great
turned off
talk.
the audio.
Hold on.
Just a second. Here. Okay, try again.
Thank you. Great talk. I think you have put some considerable thought into this this potential problem, and I appreciate that. I always appreciate somebody that actually tries to figure out something that's really wrong at the scientific level of this. So here's my take on this. It's the level question. It seems like you are talking about a very abstract dynamical system. And I almost get this feeling that it sounds like one of Eve Martyr's talks, if you understand that that reference. So I wonder if we would if you would accept for a second the premise that I'm using, that the modules of computation at the computational level of the mind would be at least relatively well characterized by neural network models that we have today. So, for example, if we were to take a model of the visual cortex, like the deep neural network models that people train up today and use for recognizing objects, you could then say what level. This would be a way to get to a concrete answer. What level of connectome information, of a static connectome information, do we need for us to recreate at a certain level of fidelity a deep neural network model of the visual cortex, or a deep neural network model of language translation, or a deep neural network model of playing a game of chess. So those deep neural network models, I think, are extremely good characteristics of the types of stuff that we should be expecting. I know that I probably get pushback from a lot of neuroscience colleagues for saying that, but I think that there's some reasonable evidence that those are good models. The other set of models that I would look at are the models of memory that are attractor based. Either short-term memory or long-term memory, hippocampal memory, sequence memory. These are all attractor-based models, at least in my understanding, and they have some significant redundancy built in at the level of the connectome. And so, if you were to go outside of your very, very general, this is a dynamic system and we need validation data, and we're to look at those types of systems that I think are at least, it could be argued, are more similar to the question at hand. Do you think that what type of validation data, what type of dynamic data do you think would be necessary for a deep neural network of a vision cortex?
That's a really good question. To be honest, I've never worked with a very large deep learning network doing vision tasks, something with millions of neurons, to see could I take some trained network like that and then reverse engineer from something I see. I mean, I don't know. I mean, what do we compare in this case? What do we compare those traces with? Let's say you're saying that if we look at the visual cortex, we can pick up the things that would tell us what are the weights in a deep learning network in this case, or
Well, for example, the connectome of a deep neural network is essentially known, right? And so you could say, if we could not estimate the weights of that deep neural network to better than 10%, how would it affect its categorization on a standardized set? What you're saying is, look, we need that standardized set, and we need to know its validation as a validation data. And I'm saying, well, we could probably do that type of experiment today on a model and see how good it was. Unfortunately, there would be you're going to get very different results depending upon the learning rules and if they use dropout of synapses and stuff like that. So you can make very robust neural network models, or you can make very non-robust neural network models. You can probably make very robust models of attractor-based memory and not so robust models. To that, I would say, gee, I can drink a bottle of liquor and still find my way home. I can, people go in for brain surgery and they get part of their cortex, or a large chunk of their cortex removed, and yet they're still working. You have quantal release probability at each synapse, so there's a good possibility that every synapse doesn't fire when it should. So there's an awful lot of noise in the system, and so there's a reason to expect there would be a lot of redundancy built into the system. So I think I think you brought up an excellent possible problem with this whole endeavor of taking a static picture of the brain, and even if it does potentially have all the information, you may have just this exponential explosion of trying to figure out how to make sense of it. But I think that there's ways to, if that really is a problem, I think there's a whole set of experiments that could be targeted directly at that that we could do today.
I can't say that I disagree. I don't. I think that that there probably are things that we can do to determine just how big that problem is or isn't, and therefore what sort of data you would or wouldn't need. And maybe the validation data is just a 5-minute fMRI, and that's it. If it really is the case that all of the circuits are so well constrained by their robustness and by these homeostatic processes that are in place, etc., that as long as you can build networks that are recognizably human, and you pick up some statistical differences that make it a little more Jane Doe, that then you can build this thing and you test it with the fMRI and it looks cool, and then you can assume this is fine. That says a lot about the success criteria that you decide to put on what makes for a successful whole-brain emulation. And you can defend them by saying, well, we did these studies that tell us that this is how robust it is, and that all of these differences that we can impose on the system still make it basically the same system. And to get a different system, to go from Jane Doe to John Doe, we need to make this many changes. If you can show that kind of a difference, and you can quantify that so that you can say, yep, it's we can rely on just this simple validation, and we can constrain our model building so well, then that's fine. And I'm not against that at all. In fact, I would hope that it was that way. I really would, because I want this to succeed as much as you do, for sure. And in fact, I try to rely on the same sort of thing. You'll see this with Dong Song when he comes on for the system identification from activity. You have to do the same sort of thing. You're picking up the bits and pieces that you already kind of know how they work. Like you've got your kernels for typical neural response in CA3 or CA1, and you're trying to map parameters for that instead of trying any possible thing, right? And you know that it's going to be probabilistic, so you're going to match a type of a probability function rather than specific spikes and all that sort of thing. We really hope that that's the case. And all I'm trying to say is you can't rush ahead and make claims about we're done, we know what we need to store, until you can actually support that with this kind of study. I think you're muted in case we're trying to answer.
Yeah, I agree. I'm not sure what to say on claims. It's a difficult position because it's kind of you could definitely take this and say, well, there you go. Call us back in 50 years, and we'll let you know if this has a chance of working. I wonder what level of proof, evidence. Proof is not the right word. What level of evidence is sufficient to be making any type of claim at all? I think the answer is you don't want to make any type of claim in the sense that you know that it's going to work, but you do want to make some kind of statement that says this has more than an epsilon chance of working, to quote my friend Sebastian Sung. And so I think there's some happy medium in between, and I would just say that this is such an important question that it blows my mind that people are not working on this, because there are some low-hanging fruit here that can actually get to exactly these questions.
Yeah, that's true. But I mean, claims are sort of what it's all about, right? Because part of the reason why you made the Brain Preservation Foundation and the prize was because you were unhappy with the claim that what had been done so far to tissue in cryopreservation was good enough. So you are saying that there is a level of, you have an assumption there about what's necessary. So, we're comparing various different claims. Like, are you going to need what Alcor provides today? Are you going to need aldehyde stabilization? Or are you going to need, in addition to that, some kind of active recording data set for validation purposes, and how much of it? It's on a spectrum, right?
Right.
Anyway, let's open this up to some other questions.
Are there any on the,
Oh, I should check, yeah. Let's see. Ooh, I'm not sure if my form is working.
there was one at, the last one for this talk was at 3:34, and it was from Leslie Seymour. Does any idea of how much of this workflow can be automated initially? Not much, then more and more of it? I don't know.
That's a good point. Actually, yeah, I like that question. Because when I was thinking about, okay, after giving this talk, it's going to seem like this is so crazy impossible, because there are so many things you have to do, and you need to test, and you need to go back and iterate, et cetera, et cetera, et cetera, which of course is what you do every time you build a model. And my answer is kind of what Leslie is asking for, is yeah, even really complicated things, even difficult things, begin to seem totally tractable and not so complicated once you put a system in place. When you build a rocket and you need to take astronauts somewhere, that SpaceX test rockets and blow up all the time, but at some point you get to a point where you've got a system in place where you have some reliability that you're building good rockets, even though they're a complex thing to build. And people 60 years ago.
Live stream just cut out.
Live stream's gone.
Really?
Looks like it.
Oh, it happens every so often. I don't know what goes on. It's on now.
Oh, it's
I
gone.
did it again. Because of an error. I hope it didn't end it. Nope, it's good.
Okay.
It's good.
Yeah, okay. So no worries, you people on the live stream who didn't miss anything. I paused while the live stream was gone. I was just going to say something complicated like building a working rocket that's eventually going to transport living people is also something that might have seemed really scary for people 70 years ago to build, because it has so many parts, so many problems. But then after a while, you manage to grasp that problem, you manage to build systems around it in a workflow and testing and so forth, and it becomes doable. And that's the same way I see this problem. We have to face the actual hurdles that are there, and you shouldn't be afraid to state them and to acknowledge them. It's just that once you see them, then you can start doing something about them. Just like the possible sort of that verification, the test that we were just talking about, Ken and I, about what could you do to find out just how robust the system is and what kind of data you need to make the right model. So yeah, I think It's the right question that Leslie's asking. See, where else can the questions come from? Oh, Michael has one question.
Or maybe slightly a comment, so you asked where the cues or like possible sources of guiding knowledge for validation.
Yeah.
So you have a slide of three circles overlapping: dynamic activity, generic human brains, dynamic activity of chain dose brain, and chain dose preserved brain. As I mentioned, there's other ones too, like external data recorded about an individual, or like you were kind of mentioning interviews or something with them. I think there's some groups that like want to do like huge numbers of questions and answers to get at sort of validation sets.
That's right. Actually, in my head, see, I'm so silly. I kind of just make up terminology and I don't explain what it is. When I say activity data specific to Jane Doe, of course, the first thought is this is all neurophysiology, recording from all the neurons, right? And that's what I mean in the worst case. But that's also dynamic activity, dynamic responses is also how do you respond to a question, behavior, all this sort of stuff.
Yeah.
So that's all in that group, just at different levels.
There's a group of people that call it a life file.
Right.
Where and they just record everything.
Yeah, that's the
And
main
part of it
right?
is for validating that, yes, actually, if you're uploaded or required to preserve that's more or less you.
It makes sense to me to have at least some kind of validation at that level.
Right.
Totally.
Yeah. I'm just not real big on the whole, is that really you question?
This all makes a lot of sense if we're talking
Right.
about bringing people back in 200 years, and there's no one alive who knows this person. So how do you know if you brought back that person, or if this is?
Well, yeah, and this is something that the cryonists have done for decades.
Right.
Yeah.
The TerraSen Foundation is also really big on that. TerraSen?
Yeah, RT, right.
I have a more like ethics-based question. So you mentioned earlier when you're designing a rocket, some of them are going to blow up, and then you ideally do that before you blow up all your rockets before you put people in them. But in this case, the rocket is a person that you're trying to bring back. So where does that leave us in this?
I was actually wondering about that myself as I was building this talk. I was thinking, ooh, look at all those millions of almost Jane Does you're producing. What do you do with them? Because are you allowed to scrap them once you've realized that they're not responding quite right? I'll be honest, I don't really know what the right answer is for this.
Well,
you have to first, hopefully Ken is right, and that things are so robust that we hardly ever get into a situation like that. Even if that's not the case, maybe you can do the validation at levels where you don't have a whole Jane Doe, but you've got pieces of the brain, and these regions, if they're okay, then you never put them together until you know that all the pieces are right. I don't know.
Yeah, I don't know if you saw, but actually this week's Nature has a paper and discussion covering, bringing up the question of exactly those ethics that you're talking about in the context of a recent experiment that was announced.
The pig brains?
The pig brains, yes.
Yeah, keeping the pig brains alive outside the skull.
But kind of in response to that, there's about a page-and-a-half discussion where they talk about, okay, we're going to have to start looking at the ethics of this, at the ethics of reviving just parts of brains, whole brains, and there's, like I said, the cryonists have been, for lack of anything better to do, have been looking at these ethical questions for a long time and don't have answers.
Yeah. So there are those questions, and then like others, like there's the situation where epilepsy patients with seizures sometimes get hemispherectomies, and now the hemispherectomy doesn't mean you take out the other hemisphere, you just isolate it. And I suppose it's possible that that hemisphere is always unconscious because it's not being fed by the limbic system, it's not being fed by the thalamus, but maybe there is something going on that's still somewhat aware. And is that okay to have this piece of the brain trapped inside of the head, not interacting with the world around it?
It's almost like that Black Mirror episode when he takes the spit.
Well,
the sample.
like Ken actually mentioned in his first talk about personhood, if you take seriously the continuity question, then I can commit the perfect murder. I kill someone and then I get intubated, get enough barbiturates to make me completely isoelectric in the brain for 36 hours, and then I come back and say, I'm not the same person anymore. I didn't do that. My copy did that. That wasn't me.
Oh, well.
Yeah, I think I think that the ethics questions really, and I'm not saying about the perfect homicide ethics question, but the stuff that Randall was bringing up about having multiple partial successes and what do you do with them. I think to a certain extent, if you look at that a different way, that's like saying, you have somebody come out of brain surgery, and they're like, oh, my mouth can't work very well. Can you put me into brain surgery again? You put them under, you tweak around a little bit more, you bring them back, and their mouth works a little bit better. There is no ethical issue with that because you're working on the same person from the perspective of there is no person, but this is the data that is most associated with that person wanted to go into the future with. So I think it might be an ethical issue that is not really an ethical issue.
Well, so now you're saying that ethically we have to do everything sequentially. We can't make the process speed up by building all these models in parallel.
I guess building it in parallel would be akin to doing these tests while the person's still under anesthesia and they never get any memory of it.
Yeah, that would work, I suppose. Yeah, under anesthesia.
Or you could do them in parallel and then serialize them. Actually, add the memories.
Right.
On the blockchain.
Okay, I think we're
Let's just put it on the blockchain
here.
But don't kid yourself, bioethicists will come up with strange arguments and take them seriously.
Brains on the
Like how to make
blockchain.
the perfect, how to do the perfect work.
Although I think, other things are going to come up first, which
Oh, yeah.
we'll get into a bit more in the panel discussion because Ken was mentioning the sort of critiques he gets from neuroscientists, and I think those are very relevant. These are critiques that are much more basic than the critique of what are we going to do when we have, a million different models of you or something like that.
Well, also also be aware that there's a tendency among bioethicists, if they can't resolve the ethical problem, to say, no, don't do it at all. And
Right.
that's not the answer that we want, presumably.